I started working on this organophosphate hydrolase, and I started working nearly about 3 decades, and mainly with the intention of developing a bioremediation strategy for inactivating the nerve agents. Organophosphates, actually the esters and thiol derivatives of phosphoric, phosphoramidic and phosphonic acid, actually they are synthesized by Bayers in 1940 during Second World War to kill Jewish people en masse. And subsequently, they are also used in concentration camps, and they actually inhibit acetylcholinesterase and prevent nervous impulse propagation and leading to death. And once their usage is prevented by UN convention, actually the Russians modified them into less toxic compounds, and they are extensively used as insecticides. Now, nearly about 75% insecticides used in the world agriculture belong to organophosphates. And these are the prominent nerve agents which are synthesized and stockpiled. Even today, their elimination is the biggest problem, and incineration is the best method to inactivate them. But for fear of release of toxic and poisonous gases, the incineration is not used, and bioremediation and enzymatic detoxification is found to be the best method. With this objective, we have started working in collaboration with the Defense Research Establishment, and we have isolated a bug which actually uses them as sole source of carbon and nitrogen. And actually, because they have one enzyme which is called organophosphate hydrolase, which hydrolyzes the third ester linkage, inactivating them and reducing the mammalian toxicity tremendously. Subsequently, once this organophosphate hydrolase is tested on this, they have tested on number of nerve agents and organophosphates. In all of them, this enzyme started working and hydrolyzing and inactivating, and therefore the general name organophosphate hydrolase is given. Organophosphates are released into the environment only about 50 years ago. Before that, the soil microbiota do not know what the kind of organophosphate is, because it is the alien substrate. So evolutionary biologists, molecular biologists, they used it as a model compound to know how the new molecules evolve in response to the accumulation of various toxic residues in the environment. And they found that the organophosphate hydrolase and lactanases, which hydrolyze the coram sensing signal molecule homoserine lactone, highly structurally similar, including active site, except one loop, which is loop number 8, which is more in lactanases. Rest of the structure is highly similar. Based on the structure and functional similarity, they found that they suggested that lactanases are progenitors of organophosphate hydrolase. And this is the phosphotriesterase, because it also is known as phosphotriesterase because it hydrolyzes the triester linkage, and this is the lactanase activity. The phosphotriesterases or organophosphate hydrolases are all having the promiscuous lactanase activity also. So once with this background, I started asking fundamental questions. Where is this enzyme located? Because once you have this enzymatic capability, unless the substrate is gaining contact with the enzyme, it is not possible to inactivate it, and therefore we actually looked at where actually it is located. Our early research way back in 2009, when we found that this is the membrane associated enzyme. The next question that we asked is, how is it targeted? There are so many pathways to target or translocate the proteins across the membrane. We just wanted to know which of the pathway is responsible for targeting the organophosphate hydrolase to the membrane. We found a signal peptide which is 23 amino acids long, and we found a twin arginine motif, not Ram Ramaswamy, it is 2 arginines. Actually, if the signal peptides having 2 arginines, these proteins, they target or translocate across the membrane in pre-folded conformation. Rest of the all proteins, they translocate in a linear mode. After reaching to the destination, they fold into active form. And this actually surprised why a metalloenzyme, organophosphate hydrolase, is having two zinc ions at the active site. And why is it taking this particular route, which is designed for transporting the pre-folded proteins? And this question led us to discover, we asked several questions. We changed this twin arginines into different amino acids, and in none of them it goes into the membrane. And it clearly tells that twin arginine motif is essential for the organophosphate hydrolase to target to the membrane. Once this is done, then we have further examined the signal peptide. And if you see this, in addition to the TAT motif, which is otherwise known as twin arginine transport motif, you have a lipobox and invariant cysteine residue at the junction of the signal peptidase keyway site. And this actually gave us a hint that this could be a lipoprotein. And that actually is true, because once we mutate this, instead of existing in the membrane, it goes into the periplasm because it cannot anchor to the membrane. And that actually further led to the discovery of linkage of this invariant cysteine residue to a diacyl glycerol moiety, which actually is responsible for keeping the organophosphate on the surface of the membrane. Subsequently, what we have done is, we wanted to know the inner membrane topology. We mutated, also removed all cysteine residues and introduced unique cysteine residues at different parts of the enzyme, and linked it to malamide reagent, which is 5 kD bigger. Once this cysteine gains access to this, because it doesn't go into the cytoplasm, it is not permeable. And once the entire protein is found on the surface of the periplasmic space, then only you will find the linkage between this lone cysteine residue and the malamide, and then you get the enhanced size of the protein. All the residues which we have introduced throughout the molecule gained access to the reagent, and we determined that the entire protein is on the surface of the molecule. Then we have asked this question. TAT substrates are proteins which require large cofactor, or the proteins which cannot acquire active conformation in harsh extracellular environment. And the third one, only multi-protein complexes. This doesn't have large cofactor, and it is not going in a harsh environment. The third possibility is multi-protein complex. And to know that, we have used two independent methods. One is metal affinity chromatography. We used Deca-Hess tag at different loop regions of the protein, and also we made the affinity column using OPH specific antibodies, and then we purified using two independent methods. We found that the native mass of the protein is nearly 290 kD, and this is just 35 kD a protein. And once you purify in a native conformation, and if you run a native gel, and it is giving 290 kD, that means it must be a multimeric protein. So this is the what is it. Then we analyzed what are the proteins with which it is associating. It is associating with TON-R, TON-B, EXPB, EXPD, and outer membrane porine W. And this is the one which we used as affinity tag to purify. So all these TAN proteins are, TAN-R is outer membrane receptor. TAN EXPB, EXPD are proton motive force components, and TAN-B is energy transducer. TON-R actually receives a number of nutrients, including vitamin B and siderophores, and this outer membrane doesn't have energy. It has to depend on the energy that is generated in the inner membrane. The EXPB-D which are proton motive force components, they harness the energy, and TON B takes the energy and gets excited and reaches to the outer membrane and transduces the energy, leading to the conformational change and then pushing the substrate into the periplasmic space. This is very well characterized in all gram-negative bacteria. Why this OPH is interacting with this? If it is interacting with this, with what component of TON it is interacting? What we have done is we have taken each and independent TON components and we expressed them with the two different affinity tags, and we expressed OPH with AVI tag and biotinylated it, and we did reciprocal pull down assays using streptavidin magnetic beads and nickel NTA magnetic beads. So once we pull OPH along with this, the EXPD is also coming, and once we pull EXPD, OPH is also coming. Number of times we tried to actually further investigate it. We have now very good bioinformatician. With the help of him, we have predicted where this can be interacted, and we found that it is interacting with arginine 91 and 96 and EXPD with leucine 83 and 81, and we actually mutated those residues and generated number of variants. Out of them, only two variants were active, and we have taken these two and did again reciprocal pull down assays. Look here, only wild type is interacting, not the variant. This says that this 91 arginine and 96 arginine, which are there on the surface of the protein, are interacting with EXPD. So subsequently, we actually did number of times heterologously to express OPH and to look where it is. In wild type, it is a membrane associated protein. When we express in E. coli, it is always found in the cytoplasm. Why is it found in the cytoplasm? What is the reason? That we were not able to know earlier. Since it was part of the component, we co-expressed these two along with EXPD and OPH. See, look, now OPH goes into the membrane. EXPD and EXPB, both proton motive force components, they actually stay together, and B is not stable in the absence of D. And therefore, we expressed them all three together. Look, the entire protein goes into the membrane. So this clearly shows that OPH, which we thought that it is evolved to hydrolyze the organophosphate, is part of energy harnessing component called TON complex in gram-negative bacteria. So another protein which I said, it transduces the energy, and with that also this protein is interacting. What we have done is we have made mutations of TON mutations in E. coli and expressed the Sphingobium TON complex, entire TON complex in cytoplasm, and we tried to pull using different affinity beads. See, anti-flag magnetic beads and nickel anti-magnetic beads and streptavidin magnetic beads. In all the cases, all three proteins are getting co-purified. So this clearly suggests that OPH is part of the four-component TON complex, hitherto non-known in any of the gram-negative bacteria, which plays a very critical role in transport of nutrients from outer membrane to periplasm. So subsequently, we reconstituted the four-component TON complex in TON negative E. coli strains, and after reconstitution, we studied its growth behavior and iron uptake using radioiso labeled iron. Look here, if you have OPH, the better growth is there than the TON complex without having this triesterase as part of it. And look here, it almost doubles iron uptake into the cell in presence of OPH. This clearly says that OPH has a role, a triesterase has a role in iron acquisition, not actually in organophosphate degradation. Maybe it is a fortuitous substrate. That is what I believe now. Further experiments we have done. They said in some evolutionary studies when they did, they said it has a promiscuous weak lactonase activity. And that is why this, they said that lactonases are progenitors of this particular molecule. Because of this, I actually tried to see, because say enterobactin is the lactone, dihydroxybenzyl serine, tris dihydroxybenzyl serine lactone, and when we incubated with that, it is becoming linear. And it is also native, it is binding very clearly. Why should it bind? Why? Because Frederick siderophore is, iron binds siderophores very strongly, and once it is reached to the periplasmic space, it is not possible to release. So there should be a enzyme which hydrolyzes the lactone background, whereby weakens the link between the iron and the siderophore and releases the ferric ions, and then in reducing environment of periplasmic space, they become, they convert into ferrous ion and they get transported into the cell. And that is what our results suggest. And see, this is the secondary binding site, and this also we have predicted and we have validated, and we mutated that lysine residue which we found at the secondary anti-binding site, and it doesn't bind in the native page. If you see that the species is not there, and but the protein is active, and SPR also we have done, it binds to this enterobactin and ferric enterobactin much more strongly. And then once we see that it is binding to iron, we have also further conducted experiment. If it is existed to acquire iron, it must be part of iron regulon. So we have looked at the genetic level to see the promoter and also the fur box, and we identified fur box, and under iron limiting condition, look here, the protein is expressed more when compared to the iron sufficient condition. That means there is a physiological link between the protein and this. And we also found the IRE elements which are otherwise found in humans to bacteria, wherein apoenzymes which actually bind to it and activate the expression post-transcriptionally. So such type of things also we have identified. We have validated the IRE element, and we showed that aconitase apoenzyme is binding to the predicted IRE, and a lot of mutational experiments which actually shows that this IRE is responsible for expression of this induction of this OPH. And we also found a small RNA which is actually is unzipping the IRE element, and further work is going on this. And these are the students who have really done. This is Sunil Parthasarthi and Ramamurthy, Aparna, Hari, and these are the people who funded the research. I am now ready to take questions.